Good afternoon and good morning, everyone. Welcome to our September Science Club. I'm Jian Li, Junior Project Manager at Science and Technology Office of Embassy of Switzerland in Seoul. We are part of the Swissness Network outpost aimed at connecting Switzerland with the world's innovation hubs. Science Club highlights the Swiss-Korean cooperation in diverse fields of studies. We organized the Science Club generally in the embassy, but because of the COVID outbreak, we switched it to a digital version, which actually offers the opportunity to be connected with our partners in Switzerland. So it is our pleasure to welcome you here all. Today's Science Club will start soon with 4 speakers. Professor Florian Theringer and Professor Sang-Chul Bae will introduce the Global Research Center for Organometrics, GROM. And then Dr. Shuasha Kao and Professor Hyun-Wook Kang will discuss the research outcomes and future visions of their joint work. Then we move on to a discussion session. First, I'd like to introduce Professor Florian Theringer. Please turn on your camera and say hello to our audience.
So, hello everyone. Jian Li, thanks a lot for the warm introduction. And it's an honor and pleasure for me to join here today, the Swiss Korean Science Club. I will shortly introduce what we actually do here in Basel, and later on Professor Bae will introduce the GROM project. It's for me a very fruitful project, and we really welcome the warm and friendly cooperation with Korea, especially with the UNIS, which is a huge pleasure and a really interesting opportunity for our research. So I can maybe go quickly and share my screen. Is it correct?
Yeah? No, I'll introduce other speakers as well. Sorry. And then I'll leave the floor to you.
So okay, I'm located in Basel, Switzerland, and I'm a maxillofacial surgeon, and I'm the head of the additive manufacturing research group located at the Department of Biomedical Engineering at the University of Basel.
Yes, thank you, Professor Theringer. And our second speaker is Professor Sang-Chul Bae. He is an associate professor at the Department of Biomedical Engineering at Ulsan National Institute of Science and Technology, UNIS. He's a director of the Global Research Center for Organometrics, where with University of Basel and Wake Forest Institute for Regenerative Medicine, the center develops organ on a chip and 3D bioprinting technologies. Please, Professor Bae, turn on your camera and say hello to our audience.
Hello, everybody. Thank you for the invitation for this good webinar. And it's my pleasure to introduce the GROM to our other audience. Thank you.
Thank you. And next, our third speaker is Dr. Shuasha Kao. Having graduated as a dentist since 2017, he's now in Medical Additive Manufacturing Research Group at the University of Basel as a PhD student. He's been working on 3D printing clinical research, especially in bone defect reconstruction. And now his current research covers the development of innovative bioinks and composite biomaterials. Please turn on your camera and say hello to our audience, Dr. Kao.
Okay. Thank you so much for that really kind introduction. And once again, thank you for organizing this online meeting and putting on a platform and for us to kind of share science and technology during this COVID time. I think it's awesome for me to be able to join in this GROM collaboration. Thank you.
Thank you, Dr. Kao. And last but not least, please welcome Professor Hyun-Wook Kang. He is an assistant professor at Department of Biomedical Engineering, UNIS. His research interests include 3D bioprinting system and process to produce biomimetic cellular construct, artificial tissue or organ regeneration, regenerative medicine, body on a chip, drug delivery system, and more. Please, Dr. Kang, Professor Kang, turn on your camera and say hello to our audience.
Hello, everyone. I'm Hyun-Wook Kang working at the UNIS. It's a great pleasure introducing our works for the bioprinting and its application. Thanks for your attention.
Thank you, Professor Kang. And at the end of this whole presentations, our innovation scout, Dr. Sunny Min, will lead this discussion session and ask our speakers for the questions for you. So before giving the floor to our first speaker, I'd like to ask our participants to write their questions during the presentation by using the Q&A section on Zoom. If there are any technical issues or technical problems, then please only use the chat section so that we could help you out as soon as possible. Now, Professor Thieringer, I'll leave the floor to you.
So thanks again. I will share my screen and then we can start. So I recently got those slides to show what innovation activities we have located here. And this is actually the platform which is also available for our collaborators from South Korea, especially from UNIST, of course, regarding the GROM project. We have on the left side, you can see Professor Hans-Florian Zeilhofer. I think he's my mentor, and I have been working together with him for now nearly 20 years. And also very important, Christian Schneider, and his whole team of specialists regarding innovation. So the Innovation Office has different focus topics. They are focused on a startup program. So actually there's a huge interest that not only Swiss, but also research groups from Korea identify their unique selling point and work on founding a startup. They can support it. They offer courses for entrepreneurship. They have, and this is also part of the GROM collaboration with South Korea, an international collaboration, and they promote this kind of activities with workshops and events. So, for example, the 6th Swiss Korean Life Sciences Symposium took place. There are industry collaborations. So if you're looking for an industrial partner for your startup, they can offer you to find the right partners for you. And in Basel, in Switzerland, we have, as you might already know, a very fruitful and active life science community. So with Novartis or Roche, there are huge companies that offer actually their collaborations if you have a potential project or a product that you would like to develop together with them. And of course, the fourth part of the Innovation Office is funding. So if you're looking for funding for your projects, they can support you in finding the right funding, grants, the right funding for your projects, including also European funding schemes. Regarding the collaboration with Switzerland and Korea, I think in the presentation of Professor Baird, there will also be some slides maybe regarding the collaborations, but you see there's already a very fruitful exchange over the last years with different activities, symposia, large meetings, presentations of Korean researchers and Swiss researchers in the different countries. So there's already a very, very active exchange of knowledge and researchers. And a very interesting new platform that I would like to present quickly is CHARMED. It's called the Center for Human Advanced Regeneration, Mobility and Education Discovery. It's a quite long word, but actually it's focused on regenerative medicine. So it fits also very well in the collaboration with GROM. So on this platform, in a collaboration with Novartis, the University of Applied Sciences, the University Hospital where I'm located, the university and the ETH Zurich, there are many partners to start a very interesting project focused on regenerative medicine. My background is maxillofacial surgery, as I already said, and I will show you quickly a little bit about my motivation, why we are active in the area of regenerative medicine, and especially also focused on the collaboration with Korea. we are interested in bringing the knowledge from the workbench to the bedside, to the clinic where the medicine happens, where we do the planning of our cases and where we treat our patients. So that means we, for many years now, we have been working on different high-tech topics like intraoperative navigation, imaging, artificial intelligence, planning of surgical procedures, also focused on, for example, laser osteotomy. I will shortly also talk about this, robotic guided laser osteotomy, and a lot of other topics. And here I would like to show a project which is also somehow connected to GROM. It's called the Project Miracle, which is funded by the Werner Siemens Stiftung. It was originally funded by 15.2 million Swiss francs. And it was initiated by Professor Hans-Florian Zeilhofer, who was also one of the most important partners for funding the GROM, or from Swiss side to fund the GROM Center. And on the other hand, it's the head of our Department of Biomedical Engineering, Philipp Katam. And with this 15.2 million Swiss francs, they started a very interesting project focused on minimal invasive robotic guided laser osteotomy, cutting bone with laser. And that should be done in a very, let's say, small approach, in an approach with endoscopes, so that you don't have large, you don't need large approaches in the operating theater for your patients. And one part is also smart implants. That includes also the design of implants that can be, for example, be smart from different perspectives. That means the surface coating, they are maybe divided in different parts so that we can have small approaches to reconstruct the human skull. But it could also be in any other case, in any other area of the human body, of course, and focus not only on bone, but also on cartilage, for example, or soft tissues. So the idea is that you can, for example, design specific geometries, always centered on the patient. That means patient specific, but also, for example, smart in regards of, let's say, sensor equipped implants. So a sensor can tell you how the implant behaves in the human body. Or another idea is, of course, that we have an implant which is biological. So that means, for example, implant that is cartilage or bone of the patient, generated for the patient. And all together is the Project Miracle. And here we see very interesting contact points also to our partners in South Korea to collaborate here. And the project that will be presented today is a part of the Miracle project, which is now again for the second phase of the project, Miracle 2, the second miracle actually, which is funded also again by the Werner Siemens Stiftung by 12 million Swiss francs. So here, and this will be shown by Professor Kang and Dr. Zhao, we are already working on a way how to regenerate patient-specifically defect areas, for example, after tumor surgery in the human body. And we are really proud to be a partner with this very, let's say, honorable, well-known research groups like Wake Forest and UNICEF, of course, an international consortium of high potential researchers to work on this very interesting topic of regenerative medicine, but also on organ mimetics. Finally, I would like to show you and invite you already, but you're also invited, of course, to come earlier. Actually, this is still under construction, but it is already, you can see the building, how it should look like here on the slides. This is our new grid campus of collaboration in Basel, in Allschwil, where our Department of Biomedical Engineering is located. You can see in my background, this is not real, it's a rendering, but still it's how this new research center will look like. And we are looking forward to be the hosts of hopefully many more Korean researchers who are interested to collaborate with us and would like to start with research in Switzerland. So thanks a lot for the invitation again, and I'm really looking forward to the rest of this session and to see what the colleagues are going to present. Thank you very much.
Thank you.
Thank you, Professor Thieringer. Now the floor is on you, Professor Day.
Okay, thank you. I will share my screen. Can you see the screen? Okay.
Yes, yes.
See that.
Okay, thank you. Again, I really appreciate this opportunity to introduce GROM to the audience. And thank you for giving me this opportunity. I thank you to the Swiss Embassy in Seoul. As Dr. Thieringer talked about, I mentioned actually the relationship started around 5 years ago with the University of Basel and UNIST. Let me briefly introduce the UNIST to the audience. UNIST is just very young university, it's just 11 years old. And we are one of the 4 research institute and also university under the Ministry of Science and Technology. And we are focused on science and technology and pursuing the excellency in the research like other institutions. And we also pursue the strong global collaborations. In that direction, actually, I was very happy, fortunate to meet the University of Basel, the strong partner in Switzerland. Actually, the interaction started when I was working at the office, University Office of Industry Relations. There I met our partners, Professor Hans Seilhofer and Christian Schneider. And we start to talk about the collaboration in many different ways. So like he mentioned, Professor Thieringer mentioned, I start to work with the University of Basel Innovation Office and how to work together in the innovation at the university. So we started with the startup collaboration, but also expanded to the research side. And fortunately, in 2018, we were awarded from the Korean government for this GROM project. This one is the research collaboration among three different institutions. The one is in the Wake Forest in US, and another partner is University of Basel and UNIST. This is the very unique opportunity for us to work with. And what is the GROM? GROM is one of the global research development center in Korea, which are funded by the National Research Foundation of Korea. Currently, around 20 centers are inactive, and they are awarded around 500K per year. And the main objective of these centers are establishing and operating joint research centers in Korea in partnership with outstanding overseas research institutions. Actually, we are the only GRDC center in Korea working with 2 different institutions, mostly the one-on-one collaboration, but our GROM is the trilateral collaboration happening. And we are very proud of that. And for the GRDC GROM project, actually, we had to focus on our own research specific areas, and we decided to start work on the organ on a chip project. What is organ on a chip? Organ on a chip is a memory stick size 3D biochip, which contain living human cells mimicking microenvironment of organs. So most of the biological systems the experiment and research has been done in the cell level, but they are very different in the tissue level. So this is the microfluidic chip, we can mimic the organ level systems in a very small size. And this organ on a chip will be fabricated in layers and channels, and that should be able to deliver all the signal, the molecules and all the environment like air and blood, so that the cells can interface with other living tissues as in a body. So instead of developing the species, actually we try to mimic the function of the body and the function of the organs. So we can print out in small size of the tissues in a small chip size. So in that way, we can mimic the function of the human. And we try to develop as a disease model. So for example, heart or brain, in case of the brain, the blood-brain barriers are very important in drug development and other functions of the brain. So that kind of thing. So we want to mimic some of the functions of the organs in a small size of the chip. So in that way, we can test this kind of the animal functions and organ functions. What's the purpose of the OOC, the organ on a chip? The goal is that we try to develop the template platform can be used for the drug discovery, the platform. So in that way, we can use this well-controlled the microfluidic chip for the drug discovery. And for example, instead of working with the animal model, we can mimic the lung functions, organs in a small size of the chip, and we can inject the drugs, and we can see the functions, how they work, and also we can mimic some of the functions of the organs. So that is the goal, and we can find the biomarkers, or we can test out the drugs, how they work with the organ, in the organ system. And we believe that the organ on a chip can be a game changer for the drug discovery. As you may know, traditionally the drug discovery always deal with the cell level and directly go to the animal model. As you know, that is really time consuming and also the very costly process. So you need a lot of the money and time for the drug discovery. In order to facilitate this kind of discovery, instead of working with the animal, we try to provide the platform, organ on a chip. We can start with the human cells, and we can not just a cell. Instead, we can use the small size organ, and we can test out those drugs, and we can find we can simply test the drugs, how they work, and whether they are toxic to the organ and how they working in the organs. So that is the main goal. And in this organ on a chip, many people is working, but there are some issues with organ on a chip, because this is really labor intensive to generate multiple organ chip in a single chip. And also it's very hard to generate a consistent organ chip. So in this technology, we try to combine the 3D bioprinting technology. So 3D bioprinting, we can print out the tissue, but in this, just the lying tissues are just the not working organ. It's just a chunk of the tissue cells. But with combining with organ on a chip, actually we can provide a micro environment like the real organ system. So, and also it's very hard to analyze the drug response in real time. So we try to combine these two, organ on a chip and 3D bioprinting technology in this center. And eventually we can use that for the drug screening platform. And so we want the different field of expertise in this development. So tissue engineering and organomimetic and drug development. For this, we want to develop the global collaboration. So UNIST, we have some experts in the biochip and also biomaterial, bioprinting. So we want to collaborate with Wake Forest. They had already the regenerative medicine. They are one of the leading regenerative medicine and also implantable organs, stem cell engineering. And the other side, also we work with the University of Basel. They are one of the top the university in the biomedical area, and they have a very good network environment over there. They are also a good relationship with global pharmaceutical companies like Roche and Novartis, and pool of the experts in the area. So they have pretty good. They are one of the top partners in this collaboration. So for that, actually, we insisted to the government. We instead of just working the one-on-one collaboration, we wanted to have a trilateral collaboration in this effort. So the GROM, our goal is to build an organomimetic drug test platform for reliable and efficient drug development. So we want to integrate all different fields of expertise in this area, and we can pursue this goal. So we want to we want to be a global R&D hub for organomimetics, and by offering a good paradigm for drug development. This is the PIs at UNIST, and Professor Hyun-Wook Kang. He is the last speaker of this webinar. He will talk about, he's an expert in the bioprinting, and Professor Chen-Young Cha, he's working in the biomaterials, and Professor Joo-Hong Kang and Taeum Park, they are the experts in the biochip area, and Chul-Min Kim in biodata, big data, and myself and the Professor Eun-Gyu Chung, we are working in the imaging, and also Hyung-Mook Kwon, he is working in drug development. So at UNIST in-house, we have some experts in these areas. And our objective is that we want to build a reliable and efficient organomimetic system. So that is our collaboration. It shows this slide shows how this trilateral collaboration working for the project. And this is a 6-year project, and phase 1 has been done last year. So we started from 2018, so we are now in the phase 2. So we established the center and the consortium and exchange programs, and we had the joint symposium last year. And unfortunately this year, because of the COVID-19, we couldn't do it offline, but we will find the way, how to continue this collaboration. In phase 2, we want to develop the core technology, and real R&D project is moving on. In phase 3, we want to find applications of this technology. So this is again the collaborators in the Basel, and the Professor Bieringer and also Professor Zeilhofer and other collaborators. And also we're working with Wake Forest University. They are one of the first regenerative medicine institute in US, and we also invite people over there. And so far, there are many meetings happen, and we visited Basel and also Wake Forest many times, and I'm very happy to see our first joint research paper published recently. And the following speakers will talk about the real research from out of this GROM project. And thank you for your attention, and I'm very happy to have questions and answers. Thank you.
Thank you, Professor Bay. Now I'll leave the floor for Dr. Cao. And because we're running a little bit behind schedule, please stick to your timeline. Okay. Now you can start sharing your screen.
Okay. Okay, firstly, let me thank the Groom project. I had a chance to do
Dr.
some.
Cao, but could you put it in the presentation mode, please?
Presentation mode, sorry.
F5.
Moment.
Or on the bottom right corner, there would be a
yeah.
icon for it.
How about now?
Perfect, thanks.
Yes, we can see.
Okay. Firstly, let me thank the Groom project. I had a chance to do some research and exchange experience in UNIST last year. This collaboration is also a great opportunity for us to take the research out into a translational medicine direction. So, as mentioned, I'm the PhD candidate and a research assistant in the University Hospital Basel and in the Department of Biomedical Engineering at the University of Basel. Today I'm talking about a little bit some of the collaborative efforts and projects that we did and currently have underway in the field of 3D printing and biomaterials, as well as bioinks fabrication. And share some thoughts we have developed on this topic. If you are interested in what we are doing in this field of 3D printing and bioprinting for regenerative medicine, I want to refer to the two web links on the bottom of this page. Okay, the oral and maxillofacial bone defects caused by trauma, tumor are common diseases causing physiological and psychological problems to the patients. With complex and architecture and unique functions in the facial and oral area, reconstructive possibilities in this field are still a challenge. What you can see here is a tumor case happened in the lower jaw. This is one of the most common maxillofacial bone defects in the clinic. Nowadays, the golden standard to reconstruct such a big defect is using the vascularized bone graft from the patient self, such as using fibula. Of course, it will cause a second injury in fibula and bring much pain for the patient. The same situation in cranial defect. Surgeons have to reconstruct the defect after brain surgery. This kind of craniofacial reconstruction are often time-consuming and present a significant challenge for the surgeons. It's difficult to carry out complex reconstruction with a precise implant, fit, and aesthetic appearance. A typical reconstruction is using a cranioplasty made by titanium or some other non-absorbable materials, which is standard neurosurgical procedures. As you may know, there is a big demand in the dental market for bone substitute. Not only the maxillofacial defect, but also the alveolar bone defect. Dentist always wants to find a good solution to repair alveolar bone defect so that they can able to to perform a dental implants in the defect area. But the commercial bone substitute made by traditional method cannot hold a well-connected porous structure, which is really important for new vessel and bone formation. Considering all the issues in the previous slides, 3D printing technology would be a promising approach in regenerative medicine. As you know, 3D printing is a really fast developing field of research since a couple of years, which now already has introduced to several applications in industrial. The upper image shows some of the most important 3D printing technologies, and all of them have been translated to the biomedical applications. In the meanwhile, like for example, the fabrication of patient-specific implants based on ceramics or metals with methods like selective laser sintering or melting. For 3D printing biological materials, only methods can be used which can be finally utilized at a room temperature, not making use of high energy of, like for example, the laser beam sintering. So we are focused on the methods which are called direct ink writing. For all the 3D printing technologies, of course, the basic principle is similar. So we need to have a virtual model and or CT scan to obtain the data from the patient, and computer added design model of the object we want to manufacture. This has to be sliced, so we have to define the layer because all the 3D printing technologies are based on a layer and a layer principle. After the slicing process, the file is more or less given to the printer. Then the real object will be generated. It's not only growing in the industrial sector, but it's growing extremely rapid in the scientific and academic field. 3D printing is especially useful in tissue engineering because, as we know, our bodies are extremely heterogeneous and complex, and 3D printing allows us to build a complex heterogeneous environment. It's widely used in tissue models for investigative biology for models that are difficult to do in vivo and currently do not have really good in virtual platforms because a lot of models, if you looking at the 2D model, are really not a good replication of the in vivo complexities. So 3D printing allows us to recreate that complexity for in vivo environment. And finally, it's a fantastic platform in personalized medicine. A large area of research and application is in the area of biomaterials, which can be defined as any material or construct that interacts with a biological system. This may be, I could say, which can be defined as this polymer, like polymers or natural polymers, as well as the composite materials, which are adapted for medical application. Now in the area of biomaterials, the most important topic are such things as strength of the material, its elasticity, porosity, solubility, whether or not it will bond to the biological substrate. that will it take or will it hold, where we want to graft to, loading force area or some small defects area. And printability, biodegradability, and very important, biocompatibility. We don't want our bodies to against the material and reject the material. We have to consider the printability of the different materials. For hard tissue regeneration, for example, ceramic materials like hydroxyapatite and calcium phosphate, they are really hard materials, can be applied in the loading force area. But on the other hand, ceramic materials are very brittle and have to be sintered after printing. The printability and degradability of polymer materials are much easier for printing and faster in degradation than the inorganic materials, which can be used for small bone defect or cartilage reconstruction. Inspired by organic and inorganic components of natural bone, polymetallic and ceramic composite is a hot topic in the area of biomaterials for tissue engineering. Because of its advantages in printability, biocompatibility, as well as mechanical property. From different clinical perspectives, we have to consider different biomaterials to meet the requirements in different applications. But still, we are facing a lot of challenges. All in all, although 3D printing is extremely useful, the simple fact of matter is that fabrication is extremely challenging. This is a lot of different factors and complexities that need to be considered before you can 3D print something and overall engineering these complexity tissues for in vivo applications. So one of the inspiration for starting this research, which is to be able to pioneer and lead the way in development of these tissues by really forming these collaborative efforts between different groups, that we have specialization in either by developing biomaterials, cell printing, or any other 3D printing technologies, as well as the idea is to bring together Swiss and Korean researchers and clinicians that will then use their complementary technologies for fabricating tissues for regenerative medicine. And another motivation of this collaboration is if you are thinking of a clinical translation, or if you are looking at GCP, good clinical practice of taking the product to the patient, you need a different set of expertise and a different level of 3D biofabrication capabilities, and it's not every academic institutes might have. So the idea of the collaboration was to really bring together these researchers to help not just develop the new advances of materials and printing technology, but also take it to the patients. As one of the outcome of this group collaboration, recently we published an article in Materials. We try to generate and completely open porous structure. The first one is polylactic glycotic acid with beta-triclosan phosphate, and and the lower material is just pure tricalcium phosphate using 2 extrusion-based 3D printing technology, as mentioned before. One is called low temperature rapid prototyping, and another one is called robotic deposition or robot casting. The printing parameters are shown on the printing parameters are shown on the right side. Then we compared these 2 3D printed scaffolds with commercialized TCP made by traditional mold forming. Another essential aspect for tissue engineering is need to functionalize the scaffold with cells before implantation. In last decades, many studies have reported that human dental pulp stem cells from the teeth has multi-differentiation potential. And like osteoblast, HDPSC expresses type 1 collagen and alkaline, as well as casein phosphate. In this study, we utilized a preliminary approach to explore the feasibility of these 2 different printing scaffolds to facilitate the reconstruction of patient-specific defects in oral and maxillofacial surgery. We expect that our study could increase the understanding of specific dental and maxillofacial applications of 3D printed polymer composite and tricalcium phosphate scaffolds in the field of hard tissue regeneration. Here are some results from this study. Let's move to this result. From the scanning electronic microscope, we can see the microstructure and material surface of conventional matte TCP and 2 different 3D printed scaffold. Compared with conventional matte TCP group, the completely open and interconnected and homogeneous 3D pore structures can be observed in 3D PLGA and TCP, as well as 3D pure TCP groups. This structure is really helpful for oxygen and nutrients applied to the cells. If you have a look at the surface of the scaffold, you can see the microporous and rough structure. From the EDS result on the right side, you will see the chemical components of these 3 materials. And table 3 shows the pore size and porosities of different groups measured by micro-CT. From our previous study, it's reported that the pore size from 400 to 600 micrometers and porosity more than 60% performed a good mechanical property and biocompatibility. As you can see, all these 3 different scaffolds present very similar pore size and porosities. And as you can see, the conventional matte TCP and 3D printed TCP scaffold exhibit higher compressive strength than the 3D printed PLGA TCP scaffold because they are pure inorganic material. Interestingly, the compressive strength of 3D printed TCP scaffold increased significantly compared to the conventional TCP scaffold, more likely because of the 3D layer by layer structure. Even they are composed of the same material. Moreover, the figure A demonstrated that 3D PLG TCP composite scaffold possessed a flexible property, which is helpful for the alveolar defect. This slide shows the biological response of human dental pulp stem cells. Live HDPSC in green color attached on the surface of the scaffold, while only a few dead cells in red color were observed on day 1 and day 3. The cells are in a healthy shape after 3 days co-culturing. It means the materials and the design structure possessed excellent cytocompatibility. This complete, well-connected internal structure helps the cells to proliferate and migrate deeper inside the scaffold. We applied Alama blue to evaluate the biocompatibility up to 7 days co-culturing.
But Dr. Cao, because we still have another speaker behind and we're running out of time, could you please wrap it up as fast as you could? Thank you.
Yeah. Up to 7 days. However, the results indicate that the cell's activity in 3D printed PLGA TCP group was significantly lower after 3 days co-culturing. Another result is osteogenesis activity of ALP. From this result, you can see that 3D printed scaffold enhanced the expression of ALP activity compared to the other two groups. Of course, as part of GROOM, we do have a lot of other projects, collaboration. Just I cannot really talk about all these projects that we do over here. But of course, the point of this is to keep growing these collaborations to have more joint projects and put the research result towards to the clinical applications. Here is some different bioinks we developed and different biomaterials, as well as we are doing some research on gelatin-based mesacrylated bioinks. So with these examples, I want to conclude my talk summarizing that there are several strategies now available for successful 3D printing scaffold. Of course, materials and technologies have to be selected for specific needs of the tissue you want to make. So there's not one technology, there's not one type of biomaterials or bioinks, but it really has to be selected for your applications. Maybe combination of several approaches I have mentioned before will lead an even better result. For example, the combination of polymer and ceramic materials. Think about the bioinks with stem cells may be and will have especially interesting properties in my opinion. And finally, what I want to mention is the blood vessels in growth, which is really important for newborn formation. So we have to keep this in mind. We need to at least macroporous so that oxygen and nutrients can diffuse to the scaffold. And it would be even better to include vascular-like structure, but this is on topic I couldn't even mention today. With this, I want to thank my team and several grants, especially GROM. Again, I want to thank you very much for listening.
Okay, thank you, Dr. Cao, and I'll leave the floor for our last speaker, Professor Kang. Please reminded that we need to have at least 15 minutes for the discussion session, Dr. Kang. Thank you. Now you could share your screen, Professor Kang.
Okay, can you see?
Yes, thank you.
Okay. Okay, hello everyone. I'm the last presenters. I'm gonna try to continue talks our works. As I introduced my works mainly focus the bioprinting technology. Today, I'm gonna introduce my works. The title is the 3D hybrid bioprinting technology to produce human scale tissue construct. The Dr. Cao usually introduced the scaffold printings. It is usually produced the biomaterials and something biomolecules to enhance the tissue formation. In comparison between the scaffold printing, bioprinting, the main difference is that just the difference of the materials. Bioprinting usually apply that the cells, living cells into the 3 structures. They also can print with the biomaterials and biomolecules to produce artificial tissue organs. The conventional bioprinting, more exactly actually cell printing, they have some difficulties. Usually they print, the conventional printing show that using the living cells, they can construct 3 structures, such as the tube and some vascular structures. But they found that some difficulty is that using the conventional cell print technology, it is only can produce tiny and weak the hybrid structures. It had the poor mechanical properties and printable size is very limited. Also, to overcome the diffusion limitation, diffusion is one of the critical factors to make living the printed cell inside structures. Channels should be incorporated, but the structure's weakness make it difficult to incorporate the channels. In this aspect, the hybrid bioprinting technology have been introduced. That is just the scaffold, and that is the combined technology of scaffold and cell printing. This is showing that one of that target structures, showing the hybrid structures. Hybrid bioprinting using the at the same time synthetic polymers and cell-laden bio-ink. Synthetic materials to provide the physical support. By providing physical support, we can more easily control them over the mechanical properties. We also can easily incorporate the channels. Cell-laden bio-ink, that is to provide the biological environment for artificial tissue formations. The hybrid bioprinting is actually the realization, not difficulties. As shown here, the multi-cartridge model usually applied for process 3D hybrid bioprinting process. Each cartridge will deliver the different materials at the same with the conventional 3D printing. By layer process, they can produce 3D cellular construct. This page is showing our bioprinting systems. As I mentioned that for hybrid bioprinting process, we need to deliver multiple type of materials. So this system had the 6 modules and enclosed system. Enclosed system was applied to controls environment such as temperature, humidity, also the cleanliness. And this is showing the one of something the fabrication result of the scaffold, just a scaffold fabrication result. This is the printing result with the polyhammer, showing that micro scale. structure can be printed. This is showing the polyurethane. Its width have about the 50 micrometers. Also, we found that our printing system can print the various kinds of the biomaterials, including PLA, PCR, polystyrene. And one of the advantage of the bioprinting is the medical imaging data can be directly applied to bioprint data. Using the CD data or MRI data, we can produce a 3D CAD models. And based on this 3D CAD model, we can produce printing information. The printing information directly can apply to bioprinting system and can produce designed structures as mentioned by the Dr. Carl. This is showing that porous maskers. This is now applied to the regenerate face skin. And this is showing other that print result showing index finger and ear scaffold. And as I mentioned, the hybrid bioprinting systems designed to concurrently deliver not only the synthetic polymer, but also cell-laden bioink. So this is showing several that the printing result with the living cells. As shown here, this structure was produced by the PCR. This is one of the thermoplastic, biocompatible thermoplastic polycaprolactone. And we also applied the red and green color cells. As you can see, we found that using our system, we can print microstructures with the living structures together with the synthetic polymer. We also found that we can easily incorporate the channels to overcome the diffusion limitations. Based on this verification rate, we try to apply this technology to artificial tissue regeneration. First, we applied this technology to the ear reconstructions. To print that ear-like structures, we applied the ear CAD model and we designed the 3D printing information. And to prepare the bioink, we isolated the chondrocyte, the cell came from rabbit ear, and we was prepared the bioink, cell-laden bioink, and we also applied the synthetic polymer, actually the bioplastic polycaprolactone. We also applied the stack-up-shear material to produce 3D structures. I can assure that you can see the movies, but we applied using the 3 materials. We processed the layer by layer process. We delivered the polycaprolactone. We also delivered the chondrocyte-laden bioink into the designed patterns. After printing, that printed structure was implanted into the mouse. This is showing that the extraction sampling result. As you can see, after that implantation, the ear shape was well that remained. And we also found that the printed ears flexibility was well maintained in vivo, because that the mechanical property usually supported by the printed bioplastics. And we have to try to investigate the mechanical property. We try to test the mechanical property through the bending test. We test the pre-implantation, and after 1 month, we tried to compare that. Left the figure showing that the pre-implantation test, bending test result, a light figure showing that after 1 month data. We found very interesting. As you can see, as you compare the 2 graphs, after implantation, the mechanical property of that printed construct was increased. This means that we don't know what happened inside of the printed construct in vivo, but we guessed some type of tissue regenerated, formed inside the structures. Based on this study result, we tried to histological assay. We first tried to investigate the gag formation usually found in the cartilage tissue. With the increase with the implantation time, 1 month and 2 months, we found that the gag content was increased. This kind of increase of gag content was also identified the subfine or staining. As you can see here, the red color showing that the gag formation. We also found that, as you can see, white dot structures. This is showing that the lacuna formation. This is the generally found in the cartilage tissue. We also found that the type 2 collagen 2 protein was also enhanced within the printed construct. Based on these results, we concluded that using the bioprinting, the hybrid bioprinting process, we can print human ear-like size and shaped structures. It had the stable mechanical properties. Also, we can induce the cartilage formation. Second, applied that the application was the muscle regeneration. Muscle have the unique morphological characteristic. First one, it had the fiber bundle structures. And more interesting is that on the fibers, muscle cell is aligned along to the fiber to generate the force. We want to mimic this kind of the muscle, the morphological characteristics. So we design and printed that. This is showing the printed result that we try to print that fiber bundle structures, and we try to align the muscle cell along to the fibers. This is showing that induced that cell alignment. As you can see here, the cell was that successfully aligned along to the fibers. And we also found that the differentiated myotube formation was also well aligned on the structures. In vivo applications, there was implanted the root rat. The alignment was well maintained. A functional improvement was also identified through the animal study. We also applied the technology to the bone printing. This is the data. This image came from the CAD CD images. As you can see, there are bony defect. We designed the 3D structures having the bony defect, and this is showing the generated the bioprinting the information. This was transferred to the bioprinting system and printed that. Similarly, with the previous, we applied the polycaprolactam bioplastic, and to regenerate the bone tissues, we applied the human amniotic the stem cell here. We also incorporate the channels to overcomes the diffusion limitation. This is showing that in vitro that the printing result, and this is showing the bone differentiation result. As you can see, printed cell was well differentiated into the bone tissues. We also did that animal study with the printed that structures. So we applied the printed structure to the calvarial bone defect model. This is showing the print result. Although they have the different oval shape in architecture, exactly the same. We printed with the polycaprolactam and TCP, and we also applied the amniotic stem cell. This is showing that pre-implantation, is showing the after 5 months. As you can see, the printed construct is well maintained in vivo. This is showing the histological assay result. We tried to compare that bioprinted group with the no treatment and scaffold only groups. As you can see here, we found that bioprinted group has a much enhanced newborn formations. We also find that by incorporating the channels, we found the more enhanced new vascular ingrowth was identified in this in vivo study. We also try to, one of the biggest difficulties in the bioprinting technology is the blood vessels. So one of the main, the focus in our study is about the blood vessels, but main difficulty is that blood vessels is too many scales. Some blood vessels have the several centimeters and tiny, the minimum the size of the about several tens of micrometers. That was a too complex pattern also. So we first try to make that artificial capillaries, but unfortunately, although the bioprinting can have the micro resolution, but it is not possible to produce a several tens of micrometer scale with the living cells. So we did make the hypothesis if we can control the direction of angiogenesis, maybe we can make that designed capillary networks. To achieve this kind of capillary networks, we use the chemotaxis pattern. To use this one, we applied the angiogenic factor secreting cells. And this is the left and right side, we located the endothelial channels, and we hypothesized if the angiogenic factor will be secreted from the cell, and this will generate a gradient of angiogenic factor, and this will produce angiogenic factor to induce sprouting in the specific directions. By controlling the direction of the sprouting, we expect maybe we can produce design the capillary networks. This is showing the simple, the parallel, the pattern result. And this is showing the endothelial channel located in both sides. And this is showing the induced the capillary networks, artificial capillary. As you can see with the time after printing, the generating, growing that angiogenesis was we found well produced from the only left to right side. And after the day 7, we found that the new capillary was reached to the angiogenic factor secreting cell. We also found that from the compound imaging, the producer that the sprouting had the tubular structures, and we also found that secured the cell-to-cell junction within that capillary networks. We try to know, can it really control that direction of angiogenesis? So we applied a different pattern. One of the inclined, we also applied the waved pattern. As you can see here, with the inclined lines bridge patterns, we successfully induced that inclined capillary networks. We also that when you applied
Sorry to interrupt, Professor Kang, but I think we might need to start the discussion session within 2 minutes.
Okay, I will
Thank you.
shortly. Yeah, and maybe the pattern we found we can control. So we also try to find a little bit more complex with successfully achieve the bridge patterns. We did also the functional test. Can it deliver the biomolecules to the designed the capillary network? And it works. By using the dextrans, we successfully transported incorporate the dextrans. We tried to apply the technology to the hepaptic models, and we successfully induced the hepaptic like that octagonal shaped with vascular structures. And after that, we incorporated the hepatocyte, and we found that by incorporating induced the vasculature, we can much enhance the functionality of artificial hepatocyte. We also try to test that the ex vivo studies. Can the printed vasculatures can integrate with the host vasculature? This is showing the implanted structures. We found that the infiltrated blood after the day 2, and it was that the blood infiltration was enlarged, and after day 8, whole structures where that blood was infiltrated. And we also found the histologic assay, the host vasculature were connected to our implanted structures. And we also found that the flowed structure is composed of our implanted cells. Our current study usually focused on the target specific tissue structure, including blood vessel, bone, nerve, skin, muscles. Based on this result, we're gonna try to focus the composite tissue regeneration, more specifically actually bone structure with that University of Basel. As I already introduced that we are having the intrinsic collaboration with the Wake Forest and University of Basel and UNIST. University of Basel have strengthened in their scaffold printing and its clinical application. Based on our strengths, we try to develop clinically applicable the technology. Yeah, this presentation, I try to introduce the hybrid bioprinting technology and its applications. And thanks for your attention.
Thank you, all the speakers. We're going to move on to Q&A session now. We had one question that was kindly answered by Professor Bae and Professor Kim. Actually, that was about the current drug discovery efforts that's going on. And if you'd like to see, you can click on the Q&A button at the bottom of your screen, and you will see the answers. And if you have any further questions, feel free to contact us as well. And I will ask you, there are 3 questions another audience has asked. So the first one is, it's more on the current limitations of the 3D printing in medicine. Could you give us some realistic picture on that? Any examples on where it's still these development and not?
Okay, so you are questioning about the current status of the bioprinting in the aspect of the clinical applications.
Correct.
Yeah, actually, there's many hurdles. Actually, the bioprinting usually applying the cells. So without that, identify the safety with using the cells. There was not we cannot apply the clinics. So current technology usually just focused to apply the scaffold. Bioprinted scaffold. As you know that the titanium printed the structure were usually easily already applied to the clinics. I guess that the current bioprint technology that cannot have still many the difficulty in produce the complex organs, but I think the simple the tissue such as the bone or a cartilage we can apply that the clinics, but the regulation, the hurdles have still maybe take some time. So if we, in the real picture, I think we can apply that the tissue in the clinics. But another issue is actually the actual money. Basically, the cell printing takes too expensive. So if we cannot find that economical the beneficials to apply clinics in the aspect of commercialization, maybe it is not easy. So nowadays, more people try to focus that uncurable that disease for that the bioprinting applications.
I see, I see. Speaking of regulation, you just briefly mentioned in the use of those things in clinical settings. Could you tell us more on the regulation regarding 3D printing use in medicine in each country that you know
of? In Korea case, actually, I don't know exactly, but when I see the overall flow that nowadays the government try to build up the regulation, but main problem is that anyone still did not report the successful result in clinical application aspect. And another problem is that usually that the regulation just focus that exactly same product. But as you know, the bioprinting, they don't have the same structure. Right. They just focus the patient-specific structures. Right. Considering this factor, now the government try to build up the regulation and try to test, actually, I think under testing the regulations. Okay. So I think after certain times, maybe there some regulation will be built up. So I think that it will take several years, maybe.
I see. Professor Thieringer, Dr. Kao, do you have any comments on that question regarding the regulations from Swiss side?
Well, I think it's somehow comparable to Korea, actually. Regarding patient-specific implants, it is that you have individualized implants, so it's a little bit different than using standard implants in surgery, for example, where the implant needs to be certified. regarding patient individual implants, it is more or less the workflow that needs to be certified. So you have to prove from the very beginning to the end, that means sterilization and packaging, that an implant is in a let's say certified valuable status and can be implanted in the patient. Regarding bio implants, it's still a little bit different because it's really focused on the patient. So here we have also different regulations, but actually we are not yet in the status that we can implant biologically generated implants. So it's still let's say in the preclinical phase.
Right. Are there like efforts to establish that sort of that regulation scheme?
Sure. Yeah, definitely. Well, regarding the medical device regulations, there will be a huge change. Well, actually, it already happened, but it was postponed until 2021 in April, as far as I know, for the European countries, that there will be a change, and which also influences individual 3D printed implants, for example. It will be, and actually I already heard from the medical device industry that it will be a really effort, a huge effort to get your tools or your implants or whatever certified for the market. So this will be, in my opinion, somehow a kind of, let's say, innovation hurdle challenge. So we will see how it will influence, especially the area of regenerative surgery or regenerative medicine.
I see. Thank you. And the last question is on the collaboration on this in this project. So I'll ask Professor Kang and Dr. Kao. So can you elaborate on the collaboration in this project? You know, which part was done where, and which center was responsible for what, and you know, how you ended up communicating with each other and all that stuff. If you could elaborate on that a little bit.
Okay. Like last year, I did almost 1 month research fellow in South Korea, and did some experiment over there. Because we are the clinical side, we find some clinical problems and looking for the biomaterials and 3D printing technologies which can help us to move forward. So we have a 3D print lab in our department and in the printers over here, and we are developing the biomaterials and some bioinks in our lab, and with experience exchange of the material properties and the properties of bioinks. So the work done here is more like from the patient data to the model design of specific implants. And then we are working on some biomaterials and bioinks for hard tissue regeneration. Yeah.
So this project was done while you were in Korea, or you started this project while you were in Korea and then you continued? I'm sorry,
yeah.
I'm not, I wasn't sure.
Yeah, first we finding the problems. We are taking the questions to Professor Kang's lab, then print and evaluate all the materials and some studies. Then I come back to Switzerland and continue this study for the development of biomaterials and bioinks for clinical applications. Yeah.
I see. And on the softer note, in a cultural side, this is my personal question. So, what were the lessons you've learned about each center, about each other, through this collaboration?
It's quite different for me, but it's of course my pleasure to be in South Korea, because I saw the people in South Korea, they are working really hard even on the weekends, doing the research and experiment during the weekends, and we exchanged a lot of experience. Yeah, and I communicate with all the PhD students on the Professor Kang's lab. So it's really good experience for me. And I hope there will be more and Korean researchers come to our lab and have a look at what we are doing in clinic and take the problems or questions back to Korea, then develop technologies or materials back to the clinic.
I certainly hope that too. Professor Kang?
Yeah.
Actually, the collaboration actually not easy. Actually, this is that, and culture difference, as you mentioned that. However, Dr. Coward that is a great guy. He did the very hard workers actually, and actually I don't know what he communicated with my student actually. And however, I think he did that good result, and it was published in the papers. He just did the several months in. Actually, he did start his work in Swiss, and he bring here and he used our print system, and we just provided something technical supporting, and we just discussed the future direction together. So, but as you mentioned, the Swiss and Korea, the deep distances, so they is not easy to overcome. But I think that there is our strength. Is Switzerland also have that something export for bioprinting. We also export in bioprinting. We are more focused on the cell printing, and the University of Basel is more focused in that the scaffold printing and its clinical applications. So I think that we can use the both system. The character cover can use ours. We expect we also can use the system of the system of the University budget. So I think that this is part maybe more easy to collaborate between your budget and universities.
Thank you very much for these answers. I certainly hope to see a lot more collaborations in the future. Well, I think it's almost time to wrap this up. Before we all go, I'd like to tell you about what we have prepared for you in the upcoming months. As part of our Life Science Initiative program, we have an open talk with experts, industry leaders, covering personalized medicine. We will discuss current system and development in personalized healthcare and cell and gene therapy. And if any startups in personalized health and digital health are watching, we have workshops for you with the industry leaders coming up as well. If you aren't receiving the newsletters from us on a regular basis, please leave a comment with your email address, and we will add you to our mailing list for more information. You can also visit our website at www.stofficesoul.ch. You can click the QR code there. And you can also find us on social media, such as LinkedIn.
And the QR code there, yes.
And the QR code there as well. Thank you, Jian. I'd like to sincerely thank our speakers and those of you who are watching us on Zoom. I'm Sunny Min, and thank you all for watching. 감사합니다.
And before I call this September Science Club to an end, I would like to ask all the speakers to turn your camera on, and we will take a short screenshot photo to keep this in a memory.
All right.
All right, all done. Now, thank you everyone for participating, and I hope you have a great evening or great afternoon.
Thank you.
Thank you very much.
Thank you very much.
Stay well.
Bye-bye. Thanks.
Bye-bye.
Stay safe.